direct measurement of water, proteins, and lipids with depth resolution in human subjects is very important for skin-related diseases for the characterization of skincare product performance. This method, along with the subsequent analysis, leverages chemometrics to extract chemical information. The main advantage of this technique is that it allows for the clinical Raman dataset to be collected by trained instrument operators that lack technical expertise to identify, exclude, and remediate all sources of spectroscopic artifacts. The resulting dataset can then be processed to identify outliers that need to be excluded from the data prior to analysis. During the data analysis, a key challenge is the removal of the outliers and identification of the number of the key components in the dataset. The approach shown in this video leverages our prior knowledge of the clinical dataset and the chemometrics approach to successfully extract the water, protein, and the lipid with depth resolution. Demonstrating the procedure will be Liang, technicians from our CND lab.
To begin, have the subject place a marked lesion body site or control site in close contact with the imaging window of the in vivo Confocal Raman instrument. Be sure that they cover the whole window to avoid the impact of room light on imaging. Then, open the software and move the focus until a spectrum similar to the one shown here is seen. Afterwards, move the focus 10 microns away from the skin surface. Collect data for 26 steps with a 2-micron step size in the frequency region shown here using